This molecule called ATP is actually a molecule called adenosine triphosphate. And the structure of this molecule is shown over here on the right-hand side of the slide. It's important to note that this is not adenine triphosphate. The structure makes it adenosine triphosphate. I know it looks a lot like a nucleotide, doesn't it? It's got a sugar right here. It's got a base that looks like adenine. And it's got not one, but 3 phosphate groups. Phosphate groups are used in living cells for a couple of different purposes, but one of the things that is very convenient about them is that they store a little bundle of energy when they are added on to a molecule. So these 3 phosphate groups make this molecule a nice container of energy for the cell. When the outermost phosphate group is broken off, and this is the group that's broken off when ATP is hydrolyzed to ADP, when this group is broken off, 7.3 kilocalories per mole is produced. Now, there is a second molecule that is very closely related to ATP. It looks a lot like ATP, except the base is guanine, not adenine. And that one is called guanosine triphosphate, GTP. Remember, GTP is considered to be the more ancient energy currency molecule. In other words, GTP most likely evolved first as an energetic coupling molecule. ATP evolved later. You still see some reactions in cells that use GTP, but you also see a lot of reactions using ATP. So there's stored energy in ATP, a pretty good amount of it. There are 3 phosphate groups, and most of the time when ATP is hydrolyzed, we get the outermost phosphate group released, and we get those 7.3 kilocalories of energy out of it. And that energy can then be used to do work. Now, I want to take a minute and talk about what happens to this phosphate group. Now note that I've listed the phosphate group as uppercase P and lowercase I. When you see a phosphate group designated this way, the I stands for inorganic. So this is inorganic phosphate, meaning there's no carbon in that molecule. You can also see phosphate drawn as an uppercase P with a circle around it. It depends on the source material you're looking at. And of course, you can write phosphate out as PO4-. Cells use phosphate groups for a lot of different things, it turns out. Of course, the most common use is for cycling between ADP and ATP in energetic coupling. Cells break off the outer phosphate group, and then they turn around and put it back on, and then they break it off, and then they put it back on, and they break it off, and they put it back on during energetic coupling. But there's another really important role for phosphate in living cells that we should be familiar with. And that's that cells can add phosphate groups onto proteins. They can also rapidly take phosphate groups off of proteins. And remember, as we said before, anytime you add a molecule onto a protein, you're going to cause that protein to change shape. Anytime you take something off of a protein, you're going to cause that protein to change shape. And anytime a protein changes shape, it changes function. So cells use phosphate groups to change the function of proteins, to turn them on or activate them, and to turn them off or inactivate them. Now, you should be familiar with the enzymes that are involved in this phosphorylation of proteins. There's a family of enzymes called protein kinase enzymes that we've mentioned before, and it is the kinase enzymes that do the work of phosphorylating proteins and other molecules. There's another family of enzymes called phosphatase enzymes, and these enzymes do the work of taking phosphate groups off of proteins and other molecules. So kinase enzymes put it on, phosphatase enzymes take it off. Anytime you take something on or off of a protein, you're going to change its shape and you're going to change its function. But again, a lot of the phosphate that's used in a living cell is being cycled on and off of ATP. ATP is a critically important molecule in a living cell because cells cannot survive without ATP. Cells do not store large amounts of ATP. They make it as they require it. Most living cells have enough ATP on hand for a few minutes, and that's it. There is no life without ATP, so ATP has to be continuously manufactured. And of course, the manufacturing of ATP is what goes on during cellular respiration. ATP is produced during respiration, and ATP is used during energetic coupling. Now, ATP production, cellular respiration, occurs in the mitochondria of eukaryotic cells. Now, I also have listed here that some ATP is produced in the chloroplast during the light-dependent reactions of photosynthesis. And you'll notice that I've put this in parentheses, and that's because the ATP that's being produced during photosynthesis, during those light-dependent reactions, that ATP is not being produced for the energy needs of the cell. That ATP is being produced so that it can be immediately used in the Calvin cycle. The ATP that is produced by the chloroplast is not produced for the energy needs of the cell. It's produced so it can be used in the Calvin cycle to make carbohydrate. So all cells, all eukaryotic cells, have mitochondria, whether they're plant cells or animal cells. And in those mitochondria, cellular respiration is going on, and ATP is being made for the energy needs of the cell. Now, if you recall from your discussion of cellular respiration that you had in BIO312, and maybe some other courses that you've taken, the way that ATP is made during cellular respiration is through a process called oxidative phosphorylation. This is a very efficient process. You get a lot of bang for your buck with oxidative phosphorylation. In other words, for each glucose, you get a lot of ATP made. Oxidative phosphorylation. During the first part of respiration, glycolysis, and during the part of respiration called the Krebs cycle, you also get a little bit more ATP made, but it's not made efficiently. It's made through a process called substrate level phosphorylation. So a little bit of ATP gets made through substrate level inefficiently. A lot of ATP gets made during oxidative phosphorylation very efficiently. And both of these things occur in the mitochondria of these eukaryotic cells. Going back to the chloroplast and the ATP that's made during photosynthesis, that ATP is made through a process called photophosphorylation. It's made using the energy of light, the energy that comes from the sun. I've got parentheses around it because, again, the cell is not making ATP through photosynthesis in order to use it for energy. The ATP that's made in the chloroplast is going to be used to build carbohydrate. Let's switch gears here and think about how energy is stored in a cell. We've talked about the fact that a covalent bond is simply a pair of electrons that's being shared between two atoms, and that electrons are the conduits of energy. So a covalent bond is storing energy in it. It's potential energy. Organisms like human beings, heterotrophic organisms, will catabolize, particularly carbohydrate, in order to release the stored energy and make ATP. A carbohydrate like glucose, for example, has about 4 kilocalories per gram. So that's a good amount of energy stored in that molecule. Carbohydrate, like glucose, is a short-term storage molecule. It gets used quickly in a cell. It's not left alone for long periods of time, but rather turned over and broken apart so that that energy can be used to make ATP. There are other molecules that are also used for the short-term energy storage in a cell. ATP is a short-term energy storage molecule. Remember, the way a cell uses ATP is to break off the phosphate bond and release some energy, or to put the phosphate back on and store some energy. So ATP can be used for short-term energy storage. The electron carrier molecules that we've mentioned, the ones that get reduced during glucose oxidation, will also temporarily store electrons and store the energy associated with those electrons. And finally, the ion gradients that we've mentioned are a short-term storage structure for the cell. There is potential energy in an ion gradient, and that energy will be released when the ion gradient is allowed to resolve. Now, it's important to know that there's one molecule, there's a macromolecule that cells use for the long-term storage of energy. In other words, fats, which are a type of lipid, are produced by a cell and then put away by the cell, saved by the cell for a time in the future when they may be short of carbohydrate or another molecule that they normally use for short-term energy storage. Fats are a molecule that has a good amount of energy stored in them, 9 kilocalories per gram. So they are a good option, a macromolecule option for cells to store energy for the long term. Ion gradients are a source of energy. They have potential energy associated with them, and they are used by living cells as a short-term energy storage system. This is short-term energy storage. The cell is going to try to resolve ion gradients as quickly as possible. But remember, cells have the capacity to maintain gradients if they spend a little energy doing that. That's what protein pumps do. That's what active transport is all about. A cell can maintain a gradient across a membrane if it spends energy to do so. It turns out that all cells have an uneven concentration of certain solutes across them between the inside of the cell and the outside of the cell. This is what we refer to as an electrical potential. We express it in millivolts because It's brought about through uneven concentrations of ions, and ions have charge associated with them. So it turns out that all living cells have an unequal distribution of charged ions across their cell membrane, and therefore a gradient across their cell membrane. And that gradient is maintained by protein pumps. We give this gradient a name. We call it the resting membrane potential. It's a potential, it's stored energy, potential energy. Now, normally, when ion gradients are formed in a cell, the cell pretty quickly moves to resolve them, and they're resolved by having the solute move across the membrane in the direction from the high concentration side to the low concentration side until equilibrium is reached. Ion gradient energy, like all gradient energy, is released when the ions flow across the membrane. But remember, ions don't move by simple diffusion. Ions move through channel proteins by facilitated diffusion. But they too will move down their gradient. That's what facilitated diffusion is. It moves ions down their gradient from the side with the high concentration towards the side with the low concentration. And it happens through these channel proteins. So some ion gradients are maintained by the cell, creating resting membrane potential, and some ion gradients are allowed to resolve through facilitated diffusion. The last thing I want to talk about today is just a very brief review of those two critically important processes that we've mentioned today, which are cellular respiration and oxidative phosphorylation. That's the processes that make ATP, and photosynthesis, which is the process that makes carbohydrate for the organism. Now, I'm not going to go through all of the details of respiration, but I want to review the key points with you because we've spent so much time talking about respiration today. Remember that cellular respiration is simply the process that oxidizes glucose to carbon dioxide. This occurs through a long series of reactions, as we said earlier. Glucose is converted to pyruvate through a process called glycolysis. This is a 10-step process. It's quite elaborate. And during glycolysis, substrate level phosphorylation occurs, that very inefficient mechanism, and it produces a net of about 2 ATP per glucose. The pyruvate that is produced through glycolysis is then converted into a molecule called acetyl-CoA through a reaction called pyruvate processing. No substrate level phosphorylation occurs in pyruvate processing. It is a one-step reaction. It produces no ATP. The acetyl-CoA is then broken down into carbon dioxide through a series of reactions called the Krebs cycle. Substrate level phosphorylation occurs during the Krebs cycle, and again, 2 ATP are made. Very inefficient process. By the end of cellular respiration, glucose has been broken down completely to carbon dioxide. The 6-carbon molecule called glucose has been broken into a 1-carbon molecule called carbon dioxide. This is oxidation because the glucose is losing all of those electrons that are in its covalent bonds. Now, all of those electrons cannot be allowed to simply go away because the energy associated with them would be lost as heat. So instead, the cell captures all of the electrons released from glucose into what we call electron carriers. And there are two involved. There's one called NAD and there's one called FADH. Now, this is not a balanced equation. I just want you to know that when you add electrons to NAD, you get NADH. When you add electrons to FADH, you get FADH2. So don't worry about balancing this out. That's not important. We oxidize glucose to carbon dioxide. We reduce electron carriers. Remember, if oxidation occurs, reduction has to occur with it. And when anything gets reduced, when it gains electrons, it also gains protons, hydrogen ions. So NAD becomes NADH. FADH becomes FADH2. Gained electrons, gained protons. Now, this whole process does not end at this point because we still haven't made the ATP. The ATP gets made through oxidative phosphorylation. These electron carriers are now going to be oxidized. They are going to lose the electrons that they're carrying into the electron transport chain. The electrons are going to be stepped down the electron transport chain, which is made up of a series of proteins, and the energy from those electrons is going to be harvested. That energy will be used not to build a new molecule, but to build an ion gradient, a hydrogen ion gradient, in fact. The hydrogen ion gradient will then be allowed to resolve. The hydrogen ions will flow through a protein channel in a molecule called ATP synthase. ATP synthase not only serves as a channel for the hydrogen ions to flow through, but also an enzyme to build ATP. So the first part of this long series of reactions is called cellular respiration. The second part is referred to as oxidative phosphorylation. The electron transport chain is just a series of proteins embedded in the inner mitochondrial membrane, and that chain will be reduced when it receives electrons from the electron carrier molecules. The electron carriers get oxidized, the transport chain gets reduced. The transport chain passes the electrons along and pulls the energy out of them, essentially, harnesses the energy out of them, and uses that energy to make a proton gradient, to send protons across the inner mitochondrial membrane into the intermembrane space. That proton gradient has potential energy associated with it, and when it is allowed to resolve, that energy can be used to do work, to build ATP. The energy from the ion gradient will be transformed into chemical energy, into the energy of ATP. ATP synthase is the molecule that does this work for the cell. The protons flow through the ATP synthase molecule embedded in the membrane, and as they flow, they cause a region of the ATP synthase molecule to physically spin. And that mechanical motion is what can then be used to build ATP. Oxidative phosphorylation produces a lot of ATP per glucose molecule. Don't worry about this number here, this 26 number, because This number is going to vary based on the source material. Just remember that during the oxidation of glucose, during cellular respiration, only 4 ATP were made. During oxidative phosphorylation, a whole lot more ATP gets made. It's a much more efficient process. It's important that we remember where this is happening in a eukaryotic cell. On this slide, you can see a drawing that represents the inside of the mitochondria and the cytoplasm. If you follow the arrows, they're showing us glucose being converted to pyruvate out in the cytoplasm. This is that process that we call glycolysis. The bonds in glucose will start to be broken down. The glucose will be broken down or oxidized into pyruvate. Bonds are broken, electrons are released. Those electrons are going to be harvested or captured by NADH. Along the way, a couple of ATP get made. The pyruvate then moves from the cytoplasm into the matrix of the mitochondria. This is the external or outer mitochondrial membrane. This is the internal or inner mitochondrial membrane. This is inner mitochondrial membrane. The blue is called intermembrane space. Intermembrane space, intermembrane space. The orange is matrix. Once the pyruvate gets into the matrix, it will be converted into acetyl-CoA. It will be oxidized. It will be broken down into acetyl-CoA. The electrons that are released will be harvested by NADH. No ATP gets made during pyruvate processing. Next, the acetyl-CoA will go into the Krebs cycle and be broken into carbon dioxide. We've gone from glucose now all the way to carbon dioxide. This is the complete oxidation of glucose. All of the covalent bonds are broken until we're left with this one carbon molecule called carbon dioxide. During the Krebs cycle, a little bit of ATP gets made, and all of the electrons that were released from the covalent bonds are going to get harvested by NADH and FADH2. Now we're done with the oxidation of glucose. So now all of these electron carriers can move forward and into oxidative phosphorylation. They're going to donate the electrons into the electron transport chain, which is sitting right here in the inner mitochondrial membrane. The electron carriers are oxidized. They're going to lose the electrons. The electron transport chain is going to get reduced. They're going to gain the electrons. The electrons will be passed down this chain of proteins, and the energy from those electrons will be used to pump hydrogen ion into the intermembrane space. By the end, the electrons will need to be accepted by a final molecule. And of course, in aerobic organisms like you and I, that final molecule is oxygen. So the electron carriers are going to get oxidized. The electron transport chain is going to get reduced. Then the electron transport chain is oxidized. The oxygen gets reduced. The oxygen is the final electron acceptor. And as it accepts the electrons, it will also take on hydrogen, follow the proton. So the oxygen will become water. This is the end of cellular respiration. So all the way through the oxidation of glucose, all the way down the electron transport chain, until the oxygen receives the electrons and becomes water. That's it. We are finished with cellular respiration. We still have not made the ATP yet. That's the oxidative phosphorylation. the hydrogen ion gradient will be allowed to resolve. It will flow through this molecule that's sitting in the membrane. This is ATP synthase. And as the hydrogen ions come through the tunnel or the channel in the ATP synthase molecule, they'll cause a portion of the molecule to spin. And that spinning motion that kinetic energy can then be converted into chemical energy by taking a DP, adding phosphate on, and turning it into ATP. This is oxidative phosphorylation. Now, something to note, this is important. The reason we call this oxidative phosphorylation has nothing to do with this oxygen molecule. This word oxidative refers to oxidation reduction. This is all a long, long series of oxidation reduction reactions that ultimately produce ATP. The final step is a phosphorylation. We're going to phosphorylate ADP and make ATP. That's why we call it oxidative phosphorylation. All cells use cellular respiration to make ATP. All of them. It doesn't matter if they're prokaryotic or eukaryotic. We've been talking about eukaryotic cells. But remember, all cells use respiration to make their ATP. They just do it differently. The bacteria and the archaea don't have mitochondria, so they can't do it the way the eukaryotes do. They still break down carbohydrate, oxidize carbohydrate in order to make ATP. They still have an electron transport chain. They still have ATP synthase. But their electron transport chain and their ATP synthase are located in the plasma membrane. So when they build their hydrogen ion gradient, they're building it across the plasma membrane. The other thing to note about prokaryotes, bacteria and archaea, is that they are much more metabolically diverse than we are. Some of them can use carbohydrates other than glucose. Everybody can use glucose. Every cell on the planet can use glucose. But some prokaryotes can also use other carbohydrates. Some prokaryotes use oxygen as their final electron acceptor, just like you and I do. But some of them are able to use other molecules, not always oxygen. They're much more metabolically diverse than we are. I want to say a few words about fermentation. Remember that for a cell that relies on cellular respiration and oxidative phosphorylation, not having their final electron acceptor available is a big problem. Organisms like human beings who find themselves without an oxygen supply are in trouble because they cannot perform cellular respiration and oxidative phosphorylation and without oxygen. The electron transport chain stops, and all of the electron carriers have nowhere to deposit their electrons. This is life-threatening. This is absolutely life-threatening. Remember, cells do not store very much ATP. They make it as they need it. So, in the short term, when your electron acceptor molecule is not available, your final electron acceptor molecule is not available to you as a cell, you can survive using an alternative pathway called fermentation. Fermentation is a completely separate pathway from cellular respiration that can produce ATP in the short term and keep a cell alive. It's going to be a way to make a little bit of ATP and it's going to be a way for the electron carriers to have a place to donate their electrons. So the electron carriers will be regenerated in fermentation, and ATP will be produced. And the reason the ATP is produced is because fermentation also involves glycolysis. Human beings and other organisms that use cellular respiration can ferment, but it's not something that we would do for the long term. We would not be able to survive using just fermentation. That's why without a supply of oxygen, a human being has a very short amount of time to live. It's important to note that prokaryotic cells also can use fermentation when they're unable to do respiration. In fact, there are some prokaryotes that use fermentation as their preferred metabolic pathway. All right, the last thing I want to talk about today is just a very brief review of photosynthesis, just to brush those cobwebs away from your memories of photosynthesis. Remember, photosynthesis is actually a combination of reactions, and we break those reactions down into two sets: light-dependent reactions and the Calvin cycle. The light-dependent reactions are the reactions that harness the light that comes from the sun and use it to build two things: ATP and NADPH. This is a different molecule than the NADH we saw in cellular respiration. So they're going to generate ATP through photophosphorylation, and they're going to generate NADPH. This electron carrier will be reduced. These two molecules will then be immediately used in the Calvin cycle. The Calvin cycle takes the products of the light reactions, ATP, NADPH, and also carbon dioxide, and it uses these to make carbohydrate, glucose. The reason that cells perform photosynthesis, if they're able, is to make carbohydrate. That's the purpose of photosynthesis. Those photosynthetic organisms will then use that carbohydrate for cellular respiration in order to make ATP. This is a diagram that puts this all together for us. It's showing us the light-dependent reactions on the top, the Calvin cycle on the bottom. It all begins with sunlight and light energy being captured by the photosynthetic cells in the chloroplasts. During the light capturing or the light-dependent reactions, ATP and NADPH will be produced. Water will be broken down into oxygen. This is the oxygen that will then be released as a waste gas. The ATP and the NADPH will then be fed into the Calvin cycle along with carbon dioxide, and carbohydrate will be produced. The energy from the sun has been transformed into the stored potential energy, the chemical energy in all of the bonds in this molecule. We see an example in photosynthesis of what we talked about at the beginning of the lecture. Cells have three choices when they're faced with a source of energy, like the sun. They can either lose that energy to the universe in the form of heat. They can use that energy in the moment and transform it into something more usable for them. Or they can put it away. They can store it for a rainy day.